We've got an interesting and fun agenda today, I like to think. This is the Leicester Life Sciences Accelerator doing their first Innovation Friday, which is quite exciting for us. We've done some virtual events before, but we've never done the sort of Innovation Friday format. Obviously, sadly lacking in samosas, but we will let you go and get your own coffee at 10 to 11. So you can even add a chocolate biscuit if you're feeling that way inclined. So we're going to start with an introduction to the University of Leicester and the life sciences capability. And it's quite a short introduction from me, a couple of slides. And then I'm going to introduce you to Dr. Tennie Videler, who's there. You want to wave and smile, Tennie? Fantastic. Tennie's had an illustrative career. She actually went and did some work in the HSN, etc., before she came back to the love of structural biology. And Tennie's the manager for the Structural Biology Institute, Leicester Institute of Structural and Chemical Biology. So some of the feedback we had at our last event was that actually people aren't really sure what does life sciences at Leicester mean. Hi, Robert. Could you possibly go on mute and switch your camera off just now? That would be great. Thank you. So I don't know how much you all know about the University of Leicester and our life sciences school, but actually we're split into 4 areas. So we've got our medical school, our School of Biological Sciences, School of Psychology, and a School of Allied Health Professions, nurses, physiotherapists, etc. And we have about 4,000 students at any one time and about 1,000 staff. So it's a really big college. For those of you in Leicester, you've probably walked past the George Davis building, past the delightful building that's the Morris Shock building. That's the big brutalistic grey building on University Road. We've got the white tiled building on the other side of the road, the Adrian building. So we're really in the centre of Leicester. And we have got lots of hospital sites as well. I'm sure some of you in the audience at some point will have been to the LRI, to the General, to Glenfield. I know I've registered them all regularly as patients. It's just been great with having small children. So the way we're split up from a research perspective is we have 2 real flagship institutes. We have the Precision Medicine Institute and the Institute of Structural and Chemical Biology, which Tennie's going to talk about today. Carl, who is one of our speakers today, Carl Edwards, is the commercial manager for LPMI as well. And we will be running an event in the future where we do a bit of a focus onto the LPMI. We've got lots of externally funded infrastructure, like you would expect any research-intensive university. But the nice thing that Leicester and our Biomedical Research Centre is known for is our chronic disease work. And at our last event, we talked about the fact that actually you've probably got the best chance in the country if you have a heart attack in Leicester. It's not to be advised, but it's always nice to know that, Glenfield's just up the road. We also have our Experimental Cancer Medicine Centre. We have a substantive site for Health Data Research UK. And one of the things that Leicester's very good at is clinical data manipulation and using that data to understand if treatments are working, what are health inequalities, developing and designing new drugs and clinical pathways. So we've got a real capability in clinical data. And at our first launch event, we talked about that capability. This is another partnership that we have that makes Leicester quite unusual. So we're partners with the hospital, with UHL, and with Leicestershire Partnership Trust. And working together, we're using all our patient data for research. So in the future, what we hope to have is a totally safe haven of data that will allow companies to ask questions and interrogate that data to accelerate product development. And then, as we've said, we've got our Biomedical Research Centre, which we focused on in our last event. And I just really wanted to say one of the things that we have been known very quickly for is our research in COVID. And actually, we've got a very large COVID study. It's £8 million from the government, called FOSP-COVID. We've also developed and delivered the first COVID recovery virtual app, which is called My COVID Recovery. So we've got a lot of expertise within the development of apps, making websites that patients can use, and linking that into people's electronic patient record. And at that, I'm going to stop and pass over to Tennie.
Hello, I'm delighted to introduce you to the Leicester Institute of Structural and Chemical Biology. My name is John Schwabe, and I'm the director of the Institute, which houses 22 active research groups, which have diverse research interests ranging from understanding the regulation of gene expression through to exploring the metabolism of formaldehyde and many other areas in between. What we all have in common is a passion for understanding the fundamental molecular mechanisms that underpin the functioning of our cells. The Institute has a number of state-of-the-art experimental facilities. Each of these houses powerful instruments that enable us to explore in exquisite detail the molecular machinery of life. This is essential for our understanding of biology and medicine. Each of our facilities is run by an expert manager, and these in their own right are outstanding scientists. A particular strength of LiSKB is the integration of structural biology with chemistry, because this allows us to develop tools which enable us to investigate and manipulate the molecular machines in our cells. You are now in our nuclear magnetic resonance facility. These NMR spectrometers allow us to study proteins and protein complexes in solution. They are particularly useful for studying protein dynamics, which is often key to understanding protein function. They are also extremely useful for studying how proteins interact with their drugs. Our three high-field NMR spectrometers, one 800 MHz and two 600 MHz instruments, are all equipped with cryoprobes that greatly increase their sensitivity. This enables us to work with especially valuable samples at lower concentrations. The NMR facility at LiSKB is one of few to be equipped with solid-state NMR probes. This allows us to study larger protein complexes and membrane-embedded proteins. LiSKB also hosts a team of scientists that develop powerful software for the analysis of NMR data. This project, called CCPN, is funded by the Medical Research Council and puts Leicester at the heart of the worldwide NMR community. Cryo-electron microscopy is revolutionizing structural biology research because it makes it possible to determine near-atomic resolution structures of the very large molecular machines that are our drug targets of the future. Our regional cryo-electron microscopy facility is a collaboration with Midlands partner universities and was supported by a multi-million-pound grant from the Medical Research Council. As well as Leicester and our Midlands partners, we host academic and industrial users from all around the world. The microscope generates vast quantities of data, as much as a terabyte each day. This is remotely accessible to our users, wherever in the world they may be. We have determined more than 35 high-resolution structures in the last 2 years. These range from fundamental machinery copying our DNA to drugs bound to their target protein complexes. As the name suggests, protein crystallography requires the molecules studied to form crystals. This is easy for small molecules, but crystallizing proteins and their complexes is often a challenge. It requires testing of many different conditions and is highly unpredictable. We have robotics so that we can test hundreds of crystallization conditions for each protein. We can even use robots to perform anaerobic crystallization trials for oxygen-sensitive proteins. We have our own X-ray generator and detector in-house so that we can screen crystals for diffraction before taking them to the National Synchrotron facility. A major strength of the Institute is that we leverage the power of chemistry to develop probes and tools to explore biological systems. Taking a chemical view to understanding biology has led to new insights, for example, into heme as a regulator of both ion channels and transcription factors, and the biological role of formaldehyde, a natural but poorly understood product of metabolism in cells. Our chemical biology researchers are developing new kinds of drugs, including PROTACs, to target gene regulatory complexes, stabilizers of protein-protein interactions, and novel metallopharmaceuticals to target cancer. Using state-of-the-art single-molecule microscopes developed within LiSKB, we're investigating complex and dynamic biological processes, including the initiation of transcription and the assembly of mRNA splicing complexes. This technology permits imaging of biological reactions at molecular scale in physiological conditions. It exploits fluorescent molecules to localize and co-localize individual biomolecules to within 10 nanometers. The microscope developed at the Institute has the ability to collect 4 colors simultaneously for extended periods of time and allows us to address questions that are beyond the reach of other methods. This includes understanding the stoichiometry and dynamics of protein assemblies, complementing the static views provided by structural biology approaches. A hub of groups specializing in single-molecule research provides the technical expertise and equipment needed to take advantage of the exciting new opportunities created by this fast-moving technology.
John has just given you the introduction to LiSKB, so I don't think I need to do that again. And it's so much nicer with beautiful pictures behind it. And I don't know why this is going on its own, but this is Cyril Dominguez, who is studying a splicing factor and has found that this is involved in cancer. So he really wants to interact with companies, especially looking for libraries to screen, again, using his assay to screen it against his targets. Another target we've got that would be of interest to pharmaceutical companies is found by Dr. Joanna Fox. And this is not a new target for cancer, but to overcome the resistance that cancer cells build up. So it's very much for dual therapy. And again, she's looking for collaborators from industry. Now, the reason I'm choosing these is because we've got more of those films, like the one that you just saw. So they're all on YouTube, and Lucy's going to send them all round. But I'm just introducing you. And really what I'm hoping is that you'll ask me questions afterwards, but these are just to give you some inspiration. Professor Vivek Gupta is a unique individual in that he's both a clinician and a structural biology expert. So as Lucy illustrated, we've got close collaborations with clinical people here in Leicester. I think it's one of our strengths, and he individually really straddles that divide. But he's collaborating with 2 companies already to look at his particular target, which is important in lung fibrosis. So he's involved in COVID research as well. But he's looking for more if anyone's interested. Spread the word. So those are targets. I just want to highlight some approaches that we can use. So Professor Mark Carr is our longest standing industrial collaborator. He's clocked up nearly 20 years, I think, of working with UCB, for example. So he looks at therapeutic antibodies, but also at how we can use antibodies to do other drug discovery. It's very exciting, and I wish I had more time to talk about this. Again, he's looking for new partners. And another example of an approach, and these people have chosen a target, but it's more about the approach. So the same with Mark. Richard Doveston looks at stabilizing protein-protein interactions. Most drugs interrupt these interactions, but there are, just like there's bad bacteria and good bacteria, there's good protein-protein interactions, and he's looking to stabilize them. He's a chemist, so he's got novel chemistry ways of doing that, and again is looking for industry partners, in this case with expertise in fragment-based drug discovery. Another chemist in the group is Dr. James Hodgkinson, and he harnesses the cell's own degradation machinery to degrade proteins that are harmful. So he's a chemist. He's very good at making PROTACs, which is a specific molecule, but he's combined his expertise, crucially, and I think this is what the Institute's good at, with the expertise of John Schwabe, who you met earlier, about histone deacetylases. So these are part of the epigenetic machinery. And so he can now target very specifically, because of the understanding of what the different histone deacetylases do. So again, they're looking for pharmaceutical collaborators, but they've already got some patents and IP, so slightly different again. So having galloped through those, I invite you to go and look at them yourself. And really what I wanted to do was invite questions from you.
Thanks, Tennie. I'm just seeing, I've asked people to put questions in the chat or to raise their hand if they have a question, but can I ask a question? Do you mind?
Oh,
so,
go on.
Say you're in the audience and listen to your talk and you think, well, so how do I interact and access LiSKB facilities? How would you go about it, Tennie?
So there are actually different ways, which is probably not the answer you want to hear, but it depends. So if you're just wanting to use a facility, you contact me or via the website, the individual facility managers, and we'll work something out. The cryo-EM, especially because it's so new, is I think there's 8 different industrial users of the cryo-EM. So Christos, who you saw in the film, he's very experienced at accommodating that, and that varies wildly from just access to the machine to collaborative projects. And same with the NMR facility. Do you just want to use, do you know how to use an NMR machine? Then we can negotiate access to it. Or do you need Fred Muscat, again, who you saw in there, expertise there? Or would you rather collaborate with all the expertise that one of our NMR groups has? So it really depends on what you're looking for.
Fabulous, thank you, Tennie. It's probably
Let's discuss.
Yeah, absolutely. And we have actually, through the Leicester Life Sciences Accelerator, started a project with a local company from Loughborough to access our CCPN.
Yeah, no, so CCPN was mentioned briefly in that. That's one of our flagship things. So Lucy just said about my misspent career. So I started off as an NMR spectroscopist, and CCPM was just a really big thing when it came along. So the fact that that is now based in Leicester, out of all the possible places it could have been in the world, to me is a source of real pride. And yes, it's great. I did not realize that Mike Bernstein was based locally, so it's really good that they're collaborating with him.
I think we're lucky to be in a heritage pharma area, and quite a lot of people in this meeting come from that environment. Anyway,
great.
Thank you very much, Tennie. What we'll do is
are there any other questions? I see on the chat. Can we share?
Alistair's asking a question.
Please do.
Alistair.
Oh, sorry, Alistair, you've got your hand up.
Yeah. Just a quick one, Tennie. Is there like a fixed price list that companies have an estimate of the cost in working with you? Do you know the time it takes to do contracts? Because that's always been the big problem. Companies come in, is actually the length of time it takes to get agreements to work. So is there any advance in that, or can you give anyone an idea of how long it would take to make such a...
Yeah, so this is really where I think Lucy probably knows more than I do, but definitely with the cryo-EM, we've got that down to a T, because I completely agree with you. That has been the problem. So since we've streamlined that, we haven't really, apart from the Amber analytics, we haven't had new users of the NMR. But I would have thought we can take that learning across. So it should be so much better than it has been in the past. If you want to try.
Angie, you've got a question.
You mute, Angie.
Obvious rookie error. Sorry about that.
You just helped me with that earlier.
I was just going to say, obviously you've had to rush through all of the elements that of your presentation because of lack of time. So are you running any, could you give us an indication of some sessions, virtual sessions and real ones that you're planning, where people interested in learning more could pop in and talk to the relevant academics or speak to you? So are you running some events that people could join and maybe you could share information about?
I think that's a really good idea. We haven't. So what we were planning to run an event last year, and we managed to convert the funding for that event to funding to make those films. So we were hoping that rather than getting everyone to be in the same place at the same time, people would take the time to go and look at these very short, beautifully made films. So I encourage people to do that. But I do think, seeing as this pandemic is taking slightly longer than everyone had hoped to, what were we thinking? We thought it would be over soon, but it isn't. So I think the time is ripe to try and do something online, because asking people about their science is just wildly different from watching it on a video. So thank you for that question. But go and look at the videos, guys.
Thank you.
And I'll share the links out with the collateral for this.
Joe's put something in the virtual chat. Could you run a virtual tour, e.g. with GoPro?
Well, how would that be better, Joe, than what you've just seen? So that was a sort of GoPro type tour, but then beautifully produced. But yeah, I can send you the rough the rough footage if you want. Sorry.
Fabulous. Right. Well, in the interest of time, I'd like to move on to our next session. But thank you very much, Tennie. And
you're so welcome, Lucy.
We'll no doubt we'll talk further. Thank you.
Yes.